Good day, we are Group 4 composed of Geri Apilado, Darylle Fidel, and I am Virtucio, and we are here to present Experiment 4, Membrane Action Potential. The semi-permeable membrane of a cell, which allows certain molecules and ions to pass through, is important in driving different biological processes. The process of ion transport and the potentials related to it are the interest of this experiment. Thus, the objectives of this experiment are to 1. Explain how membrane potential and membrane equilibrium facilitate ion transport across the membrane. 2. Compute membrane potential through the Nernst equation. 3. Explain the importance of membrane potentials and equilibria in biological processes. And 4. Explain voltage, current, and patch clamp techniques. The transport of ions depends on two things: the ion concentration gradient and the membrane potential. The ion concentration gradient is the unequal distribution of ions across the membrane, such that there are more cations outside the cell, while there are more anions inside the cell. On the other hand, membrane potential, denoted by psi, is defined as the difference in voltage or electric potential between the inside and the outside of the cell due to the ion concentration gradient. In simple terms, it is the energy needed for ions to cross the membrane. Thus, the work or free energy needed to move an ion against the concentration gradient can be written in terms of the membrane potential, where delta G is equal to negative NF delta psi, where N is the charge of the ion, F is the Faraday's constant, and delta psi is the membrane potential. Furthermore, since we previously defined membrane potential as the energy needed to cross the membrane, it can be described in terms of the total charge that needs to be transported. As such, membrane potential can be expressed as delta psi equals Q over C, where Q is the net unbalanced charge in the cell and C is capacitance. Capacitance in the context of a cell is the ability of the membrane to store electric charge by keeping apart opposite charges. Capacitance is described by the equation shown in this slide, where epsilon is the dielectric constant, A is the membrane area, and 4 pi d is the membrane thickness. From the equation of membrane potential and the equation from capacitance, it can be deduced that as membrane area increases, so does capacitance, which ultimately lessens membrane potential. This is because a large area will prevent the unbalanced charges gathered at the membrane boundary from experiencing repulsion, allowing more charge to be stored. On the other hand, membrane thickness is inversely proportional to capacitance, which increases membrane potential. This is because the thicker the membrane, the farther apart the ions from each other, and the weaker the attractive forces between the separated ions. Additionally, the picture shown here can also be used to answer the question, what would happen to the unbalanced negative charges if a few positive charges were moved from the inside to the outside of the cell? The answer is that the unbalanced negative charges would end up at the membrane boundary to avoid repulsion with the other negative charges present, and because the positive charges have increased outside the cell, the negative charges inside the cell are attracted to them. To help visualize ion movement and the membrane potential, let us look at this system. Here, we see that when ion channels are open, ions from areas of higher concentration will move towards the areas with lower concentration. However, since the membrane is not always equally permeable to the different ions, the charges do not balance out. Example, if only the sodium channels are open, the inside of the cell will become increasingly positive, while the outside of the cell will become negative. This also creates an electric gradient. When the diffusion gradient and the electric gradient are equal, then equilibrium is obtained. The voltage at this point is called equilibrium potential, or EA. In this figure, we can see that the equilibrium potentials have signs. These signs represent the direction of the membrane potential. Remember that the membrane potential is the energy needed to cross the membrane against the concentration gradient. So, since sodium wants to go inside the cell, positive membrane potential is needed to drive it out. For potassium, since it wants to go out the cell, negative potential is needed. Lastly, even though chloride wants to go inside, it will still need a negative potential to repel it back out the cell since it has a negative charge. From the previously shown system, we can use the Nernst equation to calculate the electrical force that balances the chemical force so that there is no net ion movement. This is called the Nernst potential, which is just the equilibrium potential previously discussed. This can be derived using the following equations. Delta G is equal to delta G chemical potential plus delta G electric potential. Delta G chemical potential is RT ln concentration of ions outside over concentration of ions inside, and delta G electric potential is negative NF membrane potential. Substituting this to the first equation and equating delta G to 0 because it is in equilibrium, then isolating membrane potential, we get membrane potential is equal to RT over NF ln concentration of ions outside over concentration of ions inside. The concept of membrane potential and equilibrium potential is important for different biological processes because the flow of electric charge serves as signals for sending messages in the body. This is called action potential and will be further discussed in the next few slides.
From what has been discussed so far, we know that ion channels facilitate the transport of ions from one side of the membrane to another. However, that does not mean that ion channels transport just any ions. In fact, ion channels are selective to specific anions or cations, and they can also regulate ion permeation through gating. Ion channels can be gated, meaning they can have two states, the open ion conducting state or the resting closed state. A triggering stimulus would cause the transition of ion channels from the resting to open through the process of activation. Ion channels can be broadly classified by the stimulus which stimulates their activity. The voltage-gated channels open in response to change in membrane potential. In the figure, the voltage-gated channel opens as the voltage becomes more negative. The voltage inside the membrane changes from negative 70 mV to negative 50 mV, indicating that the specific voltage for this channel to open and allow ions to cross is negative 50 mV. Ligand-gated channels open in response to ligand binding. In this ligand-gated channel, after the neurotransmitter acetylcholine binds to the extracellular surface of the channel opens, allowing the sodium, calcium, and potassium ions to move according to gradient. Mechanically gated or mechanosensitive channels open in response to mechanical stress, such as changes in pressure or changes in length. As ions move through the ion channels, the membrane potential of the cell shifts away from its resting potential. Remember that when the cell is at rest, the concentration of sodium ions outside the cell is greater than the concentration inside, and the concentration of potassium ion is greater inside than outside. In terms of charges, there are more positively charged ions outside the cell as compared to the inside. Typically, the resting membrane potential of a neuron is negative 70 mV. When there is enough stimulus to bring the membrane potential in the neuron to reach the threshold voltage of negative 55 mV or higher, an action potential occurs. Starting off, the voltage-gated sodium channels open. Sodium ions will move into the cell. Due to the positive charge of sodium ions, the membrane potential will become less negative and more positive. This is called depolarization. As the membrane potential continues to become more positive, the sodium ion channel closes, inactivating the channel. The change in membrane potential and electrochemical gradient allows potassium ions to move out of the cell. The membrane potential would then become more negative again, leading to what we call repolarization. Since potassium channels close slowly, the membrane potential becomes more negative than the resting potential. This is called hyperpolarization. The sodium-potassium pump would re-establish the potential gradient. More sodium ions will be pumped out than the potassium ions to make the membrane potential back to its resting potential.
Because voltage-gated ion channels remain open for only a few milliseconds, ion transport must be measured electrically using clamping techniques. In electrophysiology, clamping means to lock one of the electrical variables, voltage or current, while measuring the other in order to monitor the action potential. In the voltage clamp technique, the membrane is stabilized at a constant voltage while also recording the current delivered to achieve that constant voltage. Initially, the neuron is placed in a dish containing saline solution. One internal electrode measures the membrane potential Vm and is connected to the voltage clamp amplifier. The voltage clamp amplifier compares the membrane potential to the desired or command potential Vc. When Vm is different from Vc, the clamp amplifier injects current into the axon through a current-passing electrode to equalize both currents. The current required to maintain the equal potentials can finally be measured and recorded. This is an example of current recording with the voltage clamp method when a squid giant axon is voltage clamped to 0 mV. At first, no current is injected because the VC is the holding voltage of negative 65 mV, which is identical to the resting potential. When the membrane potential is stepped to 0 mV, a very brief capacitive current is seen. The capacitive current occurs because the step from one potential to another alters the charge separation and the electrical potential difference across the membrane. When VC is equal to 0 mV, the amplifier injects current which causes ion channels to open such that one sees an initial surge of inward current caused by sodium ions entering the cell. It should be noted that the inward current is analogous to negative current because the chosen convention is that a negative current value reflects entry of cations or leaving of anions. This inward current is followed by a slowly developing positive outward current caused by potassium ions leaving the cell, which continues as long as current is being injected into the cell. In the current clamp technique, the basic principle and system setup is similar to that of a voltage clamp, except that it holds the current constant while monitoring fluctuations in voltage. When current is not injected, the recorded voltage is the resting membrane potential, usually around negative 70 mV per neurons. When sufficient current is injected, such that the resulting voltage exceeds the threshold potential, the reading gives the characteristic peak of an action potential that we are more familiar with. The current is usually injected in the form of short pulses, which are visualized as square signals on the recordings. Ohm's law can also be applied in clamping techniques to obtain a simple relationship between the current injected, the voltage recorded, and the resistance of the membrane. However, in the context of cell membranes, it is often advantageous to replace the electrical resistance by its reciprocal, the conductance G, which reflects permeability of a membrane to an ion. Because the resting potential of cells is not zero, the voltage is subtracted by the equilibrium potential. Over the years, a more precise form of the clamping techniques was developed, which is known as the patch clamp. Patch refers to a small piece of the cell membrane that is isolated using a micropipette pulling it with negative pressure. The patch is placed in aqueous solution containing a buffer that blocks other ion channels. This gives the patch clamp the ability to measure the conductance of a single ion channel, giving it higher signal-to-noise ratio and temporal resolution. Depending on the locked variable, the patch clamp may either employ voltage clamp or current clamp methods. But regardless of which variable is locked, the patch clamp technique has different variants depending on the nature of the patch. In cell-attached recording, the cell membrane is not disrupted. It records voltage or current through a single ion channel. This is used to compare the response characteristics of different channels. In whole cell recording, the cell membrane is disrupted and the interior of the tip with the recording electrode becomes continuous with the cell cytoplasm. It records the voltage or current from the entire cell. In inside-out recording, the cell membrane is disrupted, but the patch of the cell membrane with the ion channel inside the pipette tip remains attached and viable. This is used to record the response of the intracellular part of single channels to different media. In outside-out recording, the cell membrane is disrupted and the patch of the cell membrane with an ion channel that remains active and viable is outside the pipette tip. When the cell membrane is repaired, the extracellular part of the channel is exposed to the media. This is used to study how extracellular chemical signals influence ion channel activity. Now that we have refreshed our knowledge on membrane potential, let's move on to our virtual lab, which is on experimenting with squid neuron to monitor action potentials and to identify the mode of action of a neurotoxin. First, we prepared a microelectrode by using a glass micropipette puller, then inserting a chloride-coated silver wire for the conduction of electrical current. Then, we extracted the giant axon from a squid, which is large enough so that we are able to insert our fine capillary electrode and measure electrical changes during an action potential when placed in a test chamber containing seawater. After connecting the other microelectrodes, we calculated the equilibrium potential for specific charged ions of the squid axon by measuring intracellular and extracellular ion concentrations and applying the Nernst equation. Then, we used current and voltage clamps to study the variations of electrical variables in a membrane and to monitor action potentials. And lastly, we identified the mode of action of an unknown neurotoxin by comparing its action potential to that of known ion channel blockers. Now for our results. In our squid giant axon, the membrane potential for sodium, potassium, and chloride ions at 20 degrees Celsius were each calculated using the Nernst equation. We were given the concentrations of each ion inside the lumen and outside the axon. For sodium, the extracellular concentration is greater than the intracellular concentration. Based on this gradient, sodium ions tend to enter the cell once it is sufficiently stimulated, thus making the inside of the cell more positive or contributing to a positive membrane potential. We can also say that the entry of sodium ions depolarizes the cell as it carries a positive charge inside. Based on our calculations, the electrical potential of sodium ions in the giant axon is positive 55 millivolts. For potassium, the extracellular concentration is less than the intracellular concentration. Based on this gradient, potassium ions tend to move out of the cell after stimulus, thus making the inside of the cell more negative or contributing to a negative membrane potential. We can also say that this moving out of potassium ions repolarizes the cell. Based on our calculations, the electrical potential of potassium ions is negative 75.6 millivolts. Lastly, for chloride, the extracellular concentration is greater than the intracellular concentration. Based on this gradient, chloride ions tend to enter the cell after stimulus. However, since they are carrying a negative charge, chloride ions entering the axon have the opposite effect compared to sodium ions. The membrane will be further polarized, contributing to a negative membrane potential. Based on our calculations, the electrical potential of chloride ions is negative 59.9 millivolts. Each ion concentration and resulting equilibrium contribute to the overall resting membrane potential, which is around negative 65 millivolts in the squid neuron and around negative 70 millivolts in the human neuron. It should be noted, however, that different types of cells have different resting membrane potentials due to different permeability to charged ions. For example, the cardiac muscle cell has a resting membrane potential of approximately negative 90 millivolts. To check if the giant axon is healthy, a current clamped at 10 nanoamperes was applied, then the fluctuations in membrane voltage were measured. The characteristic shape of an action potential was observed, specifically the depolarization phase, where the membrane potential changes from the resting value of around negative 65 millivolts to a positive value. Then the repolarization phase, where it returns to a negative value. And finally, a short hyperpolarization phase where the potential drops below negative 65 millivolts before returning to the resting value. Next, we use the voltage clamp technique to monitor changes in current at different clamped voltages. It was observed that there were no changes in current with the membrane clamped at negative 80 or negative 60 millivolts because these are below the threshold potential of negative 55 millivolts that is required to open sodium voltage-gated ion channels to trigger depolarization. In contrast, for negative 20 millivolts and above, an action potential was induced. It can also be seen that if a voltage equal to or higher than 55 millivolts is injected, no inward current is observed because the direction of the current is reversed, indicating that sodium ions instead moved out of the axon.
Afterwards, we were tasked to identify an unknown neurotoxin. Known compounds were observed for changes in action potential using current clamp technique. The figure shows the results of compound A. It could be observed that increasing the concentration of compound A inhibits depolarization, as indicated by the loss of depolarization phase. Because it inhibits depolarization, it indicates that compound A might inhibit the voltage-gated sodium ion channels as these are the channels involved in making the membrane potential more positive. For compound B, it can be observed that increasing the concentration reduces the repolarization phase and also increases the resting membrane potential. Since we know that potassium ions are involved in repolarization, drug B may inhibit the potassium ion channels. Lastly, compound C, at high concentrations, stayed constantly depolarized. It does not show a repolarization phase. Therefore, this indicates that the sodium channels have remained open and that compound C inhibits the inactivation of the voltage-gated sodium ion channels. When the unknown compound was analyzed, the results were found to be similar with that of compound A's action. The loss of depolarization phase indicates that unknown also inhibits the voltage-gated sodium ion channels. Later, the hospital confirmed that the patient ate pufferfish, which contains tetrodotoxin, a neurotoxin that binds a specific receptor on one of the subunits of the sodium ion channel. Therefore, our hypothesis that the unknown inhibits the sodium ion channels was correct.
In conclusion, we have learned that membrane potential is the difference in voltage or electric potential between the inside and the outside of the cell due to the ion concentration gradient. In the cell, since the membrane is not equally permeable to all ions, a chemical potential and electric potential are present. When these two are equal, the voltage is equal to the equilibrium potential, which can be calculated using the Nernst equation. Ion channels are what facilitate the transport of ions across the membrane and are classified into voltage-gated channels, ligand-gated channels, and mechanically gated channels. In particular, voltage-gated channels for sodium and potassium are responsible for the action potential in neurons. This process is important for the propagation of signals needed for cell communication. Ion channels can be studied using clamping techniques, specifically voltage clamp, current clamp, and patch clamp. From the experiment, we also learned that we can detect a neurotoxin by monitoring action potential using clamping techniques. Here are references for this presentation. Thank you for listening, and if you have any questions, please post them as a reply to this video. Advance Merry Christmas and Happy New Year!